Muhammad Umar Farooq biological video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. In this video we will talk about the electron transport chain. So here is the Kreb cycle. First of all we will talk about the Kreb cycle in the mitochondrial matrix inside. So the two membrane organelle are present is known as powerhouse. So the inner membrane and outer membrane and matrix are present inside of the inner membrane. So here is that is looking like a phospholipid bilayer structure is known as outer membrane of the mitochondria. So the inner membrane of the mitochondria contain complexes or protein complexes is known as inner membrane complexes. So here is a complex 1, 2, 3, 4 and 5 is known as Atp Synthase. So complex here is you can define that is the alpha F0 and F1 unit, and a P side here is the N side and alpha and beta subunit of the F1 side, and gamma epsilon is also present. So here is the another is known as F0 containing a electron acceptor, and that's electron accept to go inside in the matrix. Now it's important to understand one by one. Here is the name of the complexes are present is here. So here is the NAD H is used to, first of all we'll talk about the first complex is known as Nadph Dehydrogenase, because that is NADH splitting into NAD plus. So here is the succinate dehydrogenase is also present, because succinate convert into the fumarate, so that is due to the from Kreb cycle. So here is the cytochrome oxireductase and cytochrome oxidase and enzyme. So that is enzymatic globular protein are present in the mitochondrial inner membrane. So here is the NADH will convert into, basically NADH convert into NAD plus, and that's 2 electron and 2 proton will be accepted by the NADH dehydrogenase. So here is the FMN protein, which that is here is the alpha subunit and beta subunit, so containing a iron sulfur containing protein which that accept the electron and proton. So the proton will pump out from here to here outside into the matrix. So into the outer membrane between the for example inner membrane to outside. So here is the 4 electron will be pumped out due to the 1 NADH is here from here. So here is the throw the electron toward plastoquanine is the acceptor of electron is convert by the addition of 2 proton, and that's convert again by the pumping of. So that is why PQH2 is the real pumping system. So here is the electron transport chain contain many units. So here is the iron sulfur protein containing A, B, C, D succinate dehydrogenase complex is also well known, is known as complex 2. So here is the complex 2 is used for the plastoquanine. H2 is accept from succinate dehydrogenase complex 2, and that's exception will not pump the proton outside of the inner membrane of the mitochondria. So now never possible to matrix will allow to outside. So here is just you should remember about that. And no, next we will talk about the complex 2. So here is the cytochrome accept the 2 electron and 2 proton will be pumped out. So 2 electron will be pumped out from PQH2 into reduced in PQ, again will be come back into the so 2 proton again will be come from here. You can say that is the iron sulfur protein containing, here is the again will accept to reduce, I mean reduce to oxidase oxidized. So here is the cytochrome 2e 2 electron is accepted by the complex 4. So the complex 4 containing subunit 1 2 3. So electron will be move around the 1 2 3, and there's again backward by the cytochrome to accept more electron, and that cycle will be continue, and that transport chain will be continue. So here is the circular motion moving of the subunit to reduce the to produce oxidize the 2 electron, and proton is too much less inside because too much pumping out. So 4 electron is pumping out, and that electron here you can say that the excess of electron are present in the outside, so the proton gradient will be produced. It means inside is low and outside is high. So here is the electron will be lose their energy too much to go into the matrix, and here is the 2 electron and 2 proton, and half oxygen will be converted into the water. So here you can say that the 4 electron and 4 proton basically convert into the 2 water molecule by reduction of the oxygen 1 molecule, 1 molecule of oxygen. So here is the ATP synthase which attach from here F naught to go into the epsilon side to delta. Here is the delta side to transport, and here is the spinning due to the uh, exception of electron one by one to spinning and give the proton down from the osmotic gradient, and ATP will be produced. That energy will be stored in the form of ATP, adenosine triphosphate. So here is the Calvin cycle which that is produce FADH2 and NADH2. So now we will talk about the best information about the um, uh, another things which that is showing in the figure. For example, the basically glycerol 3 phosphate shuttle. So first of all we will talk about the Krebs cycle. So Krebs cycle give the succinate FADH2 will be produced from Krebs cycle. You know about that because we have already studied in the previous lecture. So here is the FADH2 FAD plus convert into FADH2. So here is the succinate will convert into the fumarate, and there's 2 proton will given to by the succinate to the FADH2. So the FADH will be converted from here to here. So this proton will be due to the basically enzyme succinate dehydrogenase enzyme using it, and that's spinning of the iron sulfur protein is ABCD subunit to contain, and proton will be moving pump out. So what do you think about that? If glycolysis is present in the outside of the mitochondria in the cytoplasm, so 2 NADH produced, and there's 2 NADH. For example, first we will discuss about the single NADH. So that's glycerol 3 glycerol 3 phosphate shuttle mechanism. So in this mechanism, the presence of enzyme is known as glycerol 3 phosphate dehydrogenase enzyme, which that release the proton, and that convert into NAD plus. And here is the cytosolic glycerol 3 phosphate dehydrogenase enzyme is present in us. Basically cytosolic mechanism which that are present in NADH in the cytosol. So that's come from here is the dihydroxyacetone phosphate are present here, and glycerol 3 phosphate 3 phosphate glycerol 3 phosphate convert from dihydroxyacetone, and that's give the glycerol 3 phosphate will be convert into FADH2 from FAD plus. So the FAD plus convert are reduced into the H2. So that cycle will be continue, and that's FAD again will be plastoquinine, which that is accept electron and farther way, and that's transport to the another complex. So this FADH2 is produce uh, 6 proton, because complex 1 is not involving um, in this shuttle mechanism. So here is the FADH2 is dehydrogenase enzyme. FADH2 dehydrogenase enzyme is used for it. So here is the table which that is easily understand about the calculation of the electron transport chain. So here first of all we will talk about the glycolysis produce 2 NADH, and it means 2 NADH from here, so it means not complex 1 is involved from here, shuttle mechanism, which that is not involved from here. So here is not involved, complex 1 is not involved, it means 0, and complex 2 is also 0, but 3 and 4 will be produce proton. So you can illustrate this diagram. So what will be happen? So here is the 3 ATP will be produced from glycolysis, and here is you can say that the 6 NADPH is produced from Krebs cycle. It means 4 proton and 2 proton, complex 1 and 3 and 4 will be give a total ATP. 15 ATP will be produced from the Krebs cycle, and 2 FADH2 which that is used for the shuttle mechanism. That is the basically glycerol 3-phosphate dehydrogenase enzyme to glycerol 3-phosphate shuttle mechanism to produce a basically 3 ATP. So Krebs cycle, if we will talk about here, is the 2 ATP will be produced, and glycolysis also 2 ATP. So you can write here, and that's total will be 25 ATP will be produced. So that's 25 is basically not complete, because we can imagine a different ATP we can be produced if shuttle mechanism is not involved. So the ATP will be produced in this manner. You can say here put the proton more if shuttle mechanism is not involved, and that's ATP, extra ATP will be produced is due to the 40 proton divided by 4 to 10 ATP. So we can add 25 ATP into with a 10 ATP to produce 35 ATP. So this is all the calculation, and that's not well known this electron transport chain, and their different book show the different mechanisms. So thanks for watching. Please subscribe this channel.